Hi dear students, today we begin our lesson with electron microscopy. By 1900, almost all the organelles had been discovered. There followed a time of frustration for microscopists because they realized that no matter how much the design of light microscope improved, there was a limit to how much could ever be seen using light. In order to understand why this is, it is necessary to know something about the nature of life itself and to understand the difference between magnification and resolution. Dear students, we are going to explain these two terms, magnification and resolution. Okay, let's proceed. Magnification. Magnification is the number of times larger an image is than the real size of the object. Let's suppose the real size of the object is 200 nanometer. Now, after magnification, whether the size of the image is 2 times, 3 times, 5 times, 10 times, 100 times, okay, it actually multiplies the size of the image. Magnification is the number of times larger an image. 2 times, 3 times, 5 times, 10 times, 100 times, million times. Okay, so electron microscope actually magnify an object up to million times. Okay, magnification is the number of times larger an image is than the real size of the image. The formula or the mathematical form of magnification is observed size of the image I divided by actual size of the image, or in other words you can write it as, if you know two of these values you can work out the third one. For example, if observed size of the image and the magnification are known, you can work out the actual size. Okay. Resolution. What is resolution? Resolution means the ability to distinguish between two objects very close together. The higher the resolution of an image, the greater they can be seen. For example, let's suppose there are two dots and the distance between two dots is, let's suppose, 400 nanometer. Okay, now light microscope can excellently resolve these 2 points. Let's suppose the distance between 2 points is, for example, 100 nanometer. 100 nanometer is very smaller. Therefore, light microscope will see both of the dot as one dot because light microscope cannot resolve them. Okay. Now, why light microscope cannot see things they are closer together than 200 nanometer? We are going to explain them in the coming lessons. Okay. With the light microscope, we cannot resolve or we cannot see things closer than 200 nanometer. Okay, if the 2 points can't be resolved, they will be seen as 1 point. Let's suppose we have 2 points and the distance between 2 points is more than 200 nanometer, then these 2 points can be seen through light microscope. Okay, if the distance between these 2 points is less than 200 nanometer, then the light microscope will observe them as 1 point because light microscope cannot resolve distance shorter than 200 nanometer. In practice, resolution is the amount of detail that can be seen. The greater the resolution, the greater the detail. Okay, for example, you might have seen images. For example, images with very high resolution how much you zoom in, it cannot lose the quality, because we can resolve it to a very larger amount and to a greater extent. The maximum resolution of light microscope is 200 nanometer. This means that if 2 points or objects are closer together than 200 nanometer, they can't be separate. It's possible to take a photograph and to magnify it, but we see no more detail. In other words, we do not improve the resolution, even though we often enlarge photograph because they are easier to see when larger. Okay. Dear students, here is the point. I would like you to pay attention over here. For example, then you will excellently understand the resolution and magnification. Look at these values. Okay, for example, 10 power minus 16, 10 power minus 14, 10 power, okay, it comes from here, 10 power 8, 10 power 6, 10 power 4, 10 power 2, 10 power 0, 10 power minus 2, minus 4, okay, and so on. So this represent the frequency this represent the wavelength lambda, wavelength of the waves. Okay, means 10 power minus 16 wavelength is very shorter. Very shorter wavelength means it has very high energy or high frequency. Frequency is very higher, 10 power 24. Okay, over here you can see that the wavelength is very high, 10 power 8, but the frequency is very small, 10 power 0. Okay, now we can see only the small range from this point to this point, means 400 nanometer to 700 nanometer. This is said to be the visible light. Our eyes can see things between this range, 400 nanometer and 700 nanometer. Let's suppose if an object is any smaller than 400 nanometer. Okay, so our eyes cannot resolve them. Our eyes cannot resolve them because they will see two of the point as one point. Even if it is higher than 700 nanometer, it is invisible to the eyes. So 400 nanometer to 700 nanometer, this is said to be visible light, and we can see between this light. Okay, now a wavelength shorter than 400 nanometer is said to be ultraviolet. Okay, ultraviolet is invisible to the eyes. Or for example, higher than 700 nanometer is said to be infrared. Infrared again is invisible to the light. Now this range, for example, from this point to this point, ultraviolet rays, X-rays, gamma rays, infrared rays, microwaves, radio waves, long waves, all these waves are invisible to the eyes, okay, or to the human eyes. Okay, only this range is visible to the human eyes. Okay, now the whole range of wavelength are said to be electromagnetic spectrum. This whole range of, for example, from 10 power 8 to 10 power minus 16, this whole range of wavelength is said to be electromagnetic spectrum. Okay, so X-rays, gamma rays, X-rays, ultraviolet rays, visible light, infrared, microwave, radio waves. Okay, we can see only visible light. Okay, for 400 nanometer to 700 nanometer. Okay, now look at here. This is the shortest visible light, which is 400 nanometer. This wave is 400 nanometer. Look at this structure. This structure is said to be a diameter of 1000 nanometer. Now if we are using 400 nanometer, it can easily be seen through light microscope because the size of the image is very larger. Now look at these structures. These structures are 25 nanometer, much smaller than 400 nanometer. Now these objects cannot be seen through light microscope. Okay, this object can be seen through the light microscope, but this image cannot be seen through light microscope. Okay, because they do not interfere with light waves. Okay, now it interfere with the light waves and they do not interfere with the light waves. Okay, the electromagnetic spectrum. The whole range of different wavelengths are called electromagnetic spectrum. Visible light is only one part of the spectrum. We observe a whole range of electromagnetic spectrum from 10.8 up to 10 power minus 16. This whole range of spectrum is said to be electromagnetic, and 400 nanometer to 700 nanometer is the visible light. The longer the waves, the lower the frequency, and vice versa. The shorter the waves, the higher the frequencies. In theory, there is no limit to short or how long the waves can be. Wavelength changes with energy. The greater the energy, the shorter the wavelength. If an object is any smaller than half the wavelength of the radiation used to view it, it cannot be seen separately from nearby objects. This point I would like to explain over here. For example, the shortest visible light to the eyes is 400 nanometer. Light microscope is using 400 nanometer. Now if you are using 400 nanometer, now if an object is any smaller than 200 nanometer, it cannot be observed through the light microscope. Okay, because it is not the capacity of the light microscope to view it. If it is any smaller than half the wavelength of the radiation used to view it, it cannot be seen separately from nearby objects. This means that the best resolution that can be obtained using microscope that uses visible light is 200 nanometer, since the shortest wavelength of visible light is 400 nanometer. Okay, so for example, 400 nanometer is the shortest visible light. Okay, using this wavelength we can see or resolve between 400 nanometer. If an object is any smaller than 200 nanometer, it will never be observed or seen through light microscope. Electron microscope. Types of electron microscopes. Now, transmission electron microscope. The transmission electron microscope, or TEM, was a type originally developed. Here, the beam of electron is passed through the specimen before being weaved. Only those electrons that are transmitted are seen. This allows us to see thin section of the specimen and thus to see inside the cell. Okay, transmission because the electron beam or beam of electron passes through the specimen and make a bright light on the fluorescent screen. Okay, they actually observe. With the help of those fluorescent screen we can see the image of the specimen we are going to observe. Okay, TEM has much higher resolution as compared to SEM, scanning electron microscope. Transmission electron microscope cannot make 3D images. Instead, scanning electron microscope can make 3D images of the specimen. Scanning electron microscope. The resolution of scanning electron microscope is not as good as it is of the transmission electron microscope, but scanning electron microscope actually scan the surface of the specimen and make 3D images of the specimen. The electron beam is used to scan the surfaces of the structure, and only the reflected beam is observed. The advantage of this microscope is that surface structure can be seen and a 3D appearance is achieved. The disadvantage of SEM is that it cannot achieve the same resolution as TEM, transmission electron microscope. Means resolution of TEM is very good and 3D images of scanning electron are very good. Means both have advantages as well as disadvantages. Viewing specimen with electron microscope. It is not possible to see an electron beam, so to make the image visible, the electron beam has to be projected onto fluorescent screen, as we talk about it in transmission electron microscope. The areas hit by electrons shine brightly, giving overall a black and white picture. Okay. To aid to the difficulties of electron microscope, the electron beam and therefore specimen and fluorescent screen must be in vacuum. Why in the vacuum? Because water boils at room temperature and the vacuum at room temperature. Okay. To aid to the difficulty of electron microscope, the electron beam and therefore the specimen and the fluorescent screen must be in vacuum. Means all the water along with the other particle must be removed. If electron collide with air molecule, they would scatter, making it impossible to achieve a sharp image. Water boils at room temperature in vacuum. So all specimen must be dehydrated before being placed in the microscope.